Before I get started, a few bookkeeping items. The presentation will begin shortly in about a minute or so by Dr. Gowtham Atluri, and will go on approximately till 1 p.m. Gowtham has consented to stay on for a bit after 1 to address any questions or dialogue that people may have. The slides and the recording will be available at the end of the presentation. Please log into your Malibu account to view them. Some of you who are on this call have asked me about what happened to our recording last month. There was a snafu. I don't know if it was a bug at my end or a YouTube bug, but I'm actually working with a speaker to get that event re-recorded, and that should be online next week. So thank you for your patience. If you've dialed in by telephone, you should have entered your audio pin. Otherwise, there's no way for me to unmute you in the event that you need to ask questions. If you have, if you're using VoIP, that is your headset and microphone, you should be good to go. No audio pin necessary. Please do make this, event, interactive. Ask questions. Gowtham has welcomed questions throughout the webinar, but he'll be the ultimate timekeeper to decide whether to defer your question or not. If you choose to ask questions via chat because you're in a cubicle environment, etc., please feel free to do so. Send me the question and I'll relay that to Gowtham. Let's see. For those, I think, okay, with that, let's go ahead and get started. Today, we are delighted to have Dr. Gowtham Atluri talk with us about mining neuroimaging data to explore brain dynamics. What is fascinating about this topic is this is the first webinar, at least within the Malibu system, that we are where we are exploring bringing computing, cloud, data analytics, etc., for this very, very important area for health. And so when Gowtham came into our network a while ago, and I found that this was the topic of his dissertation, etc., I was super excited. I prevailed upon him to give a talk. He's finished his PhD a few months ago, so I'm delighted to have him here. Dr. Atluri is a postdoctoral researcher at the Department of Computer Science at the University of Minnesota. He's currently working in the area of data mining under the guidance of Professor Vipin Kumar. His PhD thesis was in this area for mining neuroimaging data, and specifically his thesis was on developing data mining techniques for studying brain dynamics. He studied the role of brain dynamics in healthy and diseased subjects while they were resting or while they were performing a task. He's previously received his master's in technology from Indian Institute of Technology in Roorkee, and also his bachelor's in technology in 2004 from JNU University in Hyderabad. He's published extensively in this area, and today is going to be presenting some of his and his team's novel contributions in this space. With that, Gowtham, I'm going to turn it over to you.
Thank you, Sridhar. Hello all. The title that I have chosen is Mining Neuroimaging Data to Explore Principles of the Brain. I thought I would give you an overview of this area instead of just talking about my work. I thought I would weave in my contributions into this narrative, presenting a brief outline of fMRI data, how brain networks are constructed from this data, the challenges that underlie in constructing these brain networks, the kind of neuroscience questions that have been answered using these brain networks and some examples of those. And then I'll talk about dynamic connections and some of our recent work in this area, and then I'll conclude. Before we get into the meat of the presentation, I want to first show you how a brain plot looks like and how to interpret this plot. So here I'm showing you 4 grayscale surfaces. On the left here is the outer hemisphere of the outer surface of the left hemisphere of the brain. On the right here is the outer surface of the right hemisphere of the brain. This row has the inner surfaces or the medial surfaces of the brain. This is the left medial surface, this is the right medial surface. The values that are result of the analysis are often plotted on these surfaces to give us a sense of what they mean. So with that, I'll start a very brief introduction to fMRI data. fMRI stands for Functional Magnetic Resonance Imaging. It measures the activity at every location in the brain at a given time instance. A location in the brain is typically a 2 mm cube. For example, here I'm showing a cubic system where each 2 mm cube is referred to as a voxel, also called volumetric pixel. The measurements are done at every 2 seconds in fMRI. That's a typical resolution. There are faster and slower, but this is the standard resolution that is being used. And while these measurements are made, the subject can be either resting inside the scanner, or he could be working on a task. The task can be listening to music, or watching a movie or video, and things like that. Now here on the right is a visual of the fMRI scan. You can see that the colors are changing with time, which is indicative of the activity. Red is high activity, blue is low activity, and this data is coming from a real fMRI scan. This is not just an animation. Now, if you zoom into any particular location, you would see these voxels, these cubic voxels, and if you look at the activity over time, it can be represented as a time series like this. Now, all the voxels in the brain, at 2 mm resolution, there are about 100,000 voxels, and they can be represented in the form of this matrix on the left here. The x-axis is time, the y-axis are the locations or voxels, and each value indicates the activity. So this is a very high-level overview of what fMRI data is and how to visualize it. Now, there are other spatio-temporal data sets available out there. For example, this is a vegetation data. What it has is the vegetation information of every location on the surface of the Earth at every time point. Here, the data is shown at an yearly scale. So this is very similar to the brain data because for every measurement has a location and a time associated with it. This one is the crime data from Chicago. At every location in Chicago, the intensity of the crime is shown on a yearly scale. And this one is a brain image from an Alzheimer's subject. The gray colors here represent the tissue that was lost, and these scans were collected, I think, on a monthly basis. So again, the data has both location, which is in a brain, and also a time instance at which this data is collected. Similarly, traffic data is collected widely these days. The sensors that are placed on the freeways at interesting locations, they measure the traffic flow regularly. And this data is also in the public domain. So all these data sets share, including the fMRI, share this common characteristic that a measurement has both space and time associated with it. And so the problems that are studied in these domains are also very similar. And there is an opportunity here to cross-pollinate ideas between these domains. And you may be working on some spatio-temporal data sets that I have not listed here. And I'd be happy to hear what you think about the problems that we talk later on. And this studying these data sets has the potential to address society's problems in terms of climate change, health, crime, or traffic congestion, etc. Now, going back to fMRI data, one can analyze this fMRI data and can understand the brain's operating principles at rest. One can also understand how the brain responds to external stimuli, or one can understand how the healthy and diseased brains are different. Recently, in 2012, it's been 20 years since fMRI was invented, and the Neuroimaging journal, which is one of the major journals in this area, they published a special issue in which they invited reviewers from leading researchers in this area. And some of them have reflected on the success stories of fMRI. Some of them have expressed dissatisfaction on the advances that were made in this area. And others have outlined some future directions that fMRI analysis can take. Now, for a long time, we have known that the brain is a network of neurons. And so one approach to analyzing fMRI data is to model this data as a network. So this slide outlines a basic pipeline that is used to construct brain network from fMRI data. On the left here is the representation of the fMRI data. We have a time series for every voxel in the brain, but these time series have an interesting characteristic called spatial autocorrelation that I am showing you on the right side of the screen here, which means that the adjacent locations of a voxel share very similar time series. So to sort of handle this, typically a brain atlas which merges some of these voxels into bigger regions is used, and the time series of all the voxels in a brain region are used to compute the mean time series, which represents the time series for the entire region, and we get the region time series for all the regions in the atlas. Typically, there are 100,000 voxels, and the regions are of the order 100 to 200 in a typical atlas, and so we get 100 to 200 time series here. One can then construct a pairwise correlation matrix, which will be a region cross region matrix as shown here, and each element is a correlation between the corresponding regions. Now, this pairwise correlation matrix can also be treated as a weighted brain network, where each region is a node, and the strength of connection of any connection is basically the correlation between the corresponding regions. One can either.
There's a question here.
Yes, please.
The questioner asks, I guess and please jump in if I'm not representing it correctly. The purpose of converting voxels or identifying the regions, is it a computational issue, that is to say there are too many voxels otherwise, and or is it to actually model this more accurately?
I think both reasons are valid, because constructing a 100,000 network will be compute intensive, and studying it over time in multiple cases does bring in computational challenges. At the same time, there is redundancy in the data to some extent, so that can be handled, that can be reduced to sort of improve the computational requirements.
I see. Okay. Thank you.
So the weighted brain network can then be binarized to get a binary brain network. The way to do it is one chooses a threshold of correlation, and any connection whose strength is greater than the threshold is treated as an edge in the binary network. Otherwise, that edge is dropped. Now, this is the pipeline for constructing a brain network using a brain atlas. There are also other ways of constructing these brain networks, which is they reduce the resolution of the data by going from 2 mm to 3 mm or 4 mm. That way, they reduce the number of voxels from 100,000 to roughly 40,000 or so, and then they compute the pairwise correlations, and they get the weighted brain network. And this can further be binarized, and so a voxel level network can also be constructed. The idea here is that once we have these brain networks, we can study the properties of the brain and understand how it works. Now, through this pipeline, there are some choices that have been made, and those choices will have consequences. So I'll outline a few of those choices that could play a major role in the analysis. For example, here, Zaleski and colleagues, they studied the impact of the number of regions that are chosen in the analysis on the results of on the outcome of the analysis. So they looked at this map shown here, which has 82 nodes in the standard anatomical atlas. Each region is shown in a different color here. They then broke each of these regions into smaller fragments, and they ended up with, their goal was to get 500 nodes. They further split these nodes into 1,000 nodes, and so on until 4,000 nodes. So they have 6 different sort of atlases at different granularity. Now, one can construct a brain network using these different atlases, and then one can study its properties. In this case, they looked at the degree of the network. Here they are showing the degree of the network in different subjects. So this is subject 1, this is subject 2, and here they are showing the average over a population. And each row is one particular atlas. This is the 82 node atlas, this is the 500 node, 1,000, and 3,000. Now, if you look at this average map here, you see, and the color indicates the degree, and white is higher, darker color is lower degree. And in this map here on the right, you see that the white regions are mostly at the rear of the head up here. And then there are other guys that are the other locations that have reasonably higher degree as well. As you go down this column, you see that the color of the map is changing drastically, and also the location of the high degree nodes is changing too. For example, here it's high here, but in the next one it's high somewhere in the middle, same here, and then it becomes smaller and smaller. So what they're trying to say here is that the number of regions that are chosen to construct the brain network will have an impact on the outcome of the analysis.
Gowtham?
Yes.
A quick question. I might have missed this. What is the degree of a region?
I'm sorry, the size?
What is the degree? I mean, how are you defining what a degree is?
Good question. I'm sorry, yeah. So the degree of a region is basically the number of other locations that a region is connected to. For example, here, although it's not clear, let's say this region is connected to 4 other regions. So the degree of that region would be 4. So it's how well a node is connected to the rest of the network in terms of its edges.
I see. So this degree then just corresponds to sort of modeling this as a graph and then viewing it as a degree of a particular vertex.
That's right. Yes.
Thank you.
And then there are choices often made in terms of the atlas that is used. In the previous slide, we saw the impact of the number of nodes. Here, these are different atlases that came out of different labs. So the left is the Harvard-Oxford Atlas, the middle one is the Tallerac Atlas, and this one is the MNI, I think it's Montreal Neuroimaging Institute Atlas. So these can have an impact of the analysis too, because if in one atlas, if this is the region that was indicated, and if a different atlas chooses that as a region, now they are slightly different, and so the time series that is averaged based on the locations can look different, and so the correlations can change too. So the choice of atlas could potentially have an impact on how the network would look or the kind of results that we would get at the end of the analysis.
Gowtham, one more question.
Sure.
The questioner asks, are the regions related to the function that a particular region in the brain performs, or is it somehow related to, let's say, what a particular diseased patient might be experiencing?
I think that's a very good question. So the regions are determined in many different ways. One of the ways it is determined is by looking at the lobes in the brain. So if you look at the brain, there are these folds, and based on these folds, any tissue or any gray matter that is present between 2 folds is often treated as a region. There are other ways to determining regions. Sometimes they look at the type of cells that are present in the tissue in these places. And by looking at these cells that are similar, they hand carve these regions. So sometimes they do it in a data-driven fashion too. One way to do it using fMRI data could be to cluster these time series and look at, and based on that determine where the regions are that have very similar time series. So there are a plethora of ways in which the regions can be determined. And there are atlases that are out there that come from each of these different categories.
Thank you.
Now, the other potential factor is the length of the scan that is used to construct the brain network. To study this, Anderson and colleagues from University of Utah, they collected 55-minute resting state scans from one subject. Here in the left, what they did is they took one 5-minute scan, they computed the brain network or the correlation matrix, and they took another 5-minute scan, they computed the correlation matrix, and then they asked the question, how similar are these two brain networks? And this scatter plot is drawn to answer that question. As you can see, the correlations are in a cloud. There is no sort of linear relationship that you can see in this cloud. So this plot suggests that if you use a 5-minute scan, the correlations are unreliable. On the other hand, they combine 5 different scans to get a long 25-minute scan. This is not one scan, but this is a, appended 5-minute scans. And then they computed the correlation matrix. They repeated this with a different set of 5 scans. They again computed the correlation matrix, and then they compared the correlation values. Here you see that they are much more linearly aligned than in the previous case. The basic message from this paper is that if you use a 25-minute scan, you can get a more reliable network from a single subject. But if you use a scan that is longer than that, the value addition is not much. So this basically sort of shows that if depending on the length of the scan, the network could look different, and the reliability can also be very different. Another potential factor is motion that is there when a scan is obtained. Subjects often move their heads in the fMRI machine when the scan is obtained, just because it's very inconvenient. I don't know how many of you have been in an MRI machine. It's often noisy, it's very congested, and so on. So here, what they are showing in this analysis, Power et al., this paper came out in 2012, they considered 3 regions at the back of the head. And here they are showing the fMRI time series from these 3 different locations. And here in the bottom plot, they are showing the change in the head position in red color. Now, if you notice, wherever there is a big change in the head position, there is actually a big change in the values in the fMRI time series. This suggests that this is perhaps not real activation, but this is because of the change in the position. And so if we construct correlations based on these values, the correlations could be spurious. So what they suggested is to drop the time frames wherever the change in head position is very high. And here in the bottom, the brain plot here, they are showing the edges in blue color are the edges that are filtered out by dropping these time frames where there is a lot of change in head position. And in red color, they are showing the connections that surface because we have dropped these connections. So, and then as you drop more and more frames where there is more motion, you see that there are more and more blue connections that come up at the top of the head, and there are a lot of connections from the rear to the front of the head that surface. So they are showing that as you address these motion problems, you can get to real connections, or hopefully real connections. So in the last few slides, I have shown that there are several factors that could affect the brain network and also the downstream analysis. Now, let's look at the neuroscience questions that are studied using these brain networks. So some of the questions are, what are the principles of the brain networks when the subject is resting? That is, what are the characteristics of these networks when the subject is resting, and how do these characteristics change when the subject is presented a stimulus? The other question could be, how are these brain networks changing as the person goes from childhood to youth to adulthood? A related question could be, as the person is learning a skill, how does the brain network change accordingly? And perhaps a more important question is, what are the differences between healthy and diseased brains? Excuse me. Answering these questions will help us understand the causes for these mental disorders, as well as design treatment methodologies to treat subjects with these disorders. Now, here are the relevant data mining tasks that pertain to the earlier questions. What are the non-trivial characteristics of these brain networks? When the subject is resting, are the brain networks looking like random networks? Are there any non-trivial characteristics that make them stand out? Examples are the presence of hubs or high clustering coefficient. Many of these characteristics were first studied in social networks and also in a plethora of other networks, including biological, web graphs, co-authorship networks, transportation networks, etc. A related question is, what are the types of structures that these networks have? Are there communities in these networks? Are there motifs like the star structure that is shown here? Or are there bike-leaf structures in these networks, and what does that mean? And these questions can also be asked when comparing 2 different networks. One could compare a healthy network with a diseased network and ask, are the edges different? Are there differences in the characteristics of these networks? Are the structures that are present in one network absent in the other, or vice versa? And so on. We have done some work in finding some interesting bike-leaf structures like these that are present in schizophrenia subjects but that are absent in the healthy subjects. And I'll cover some of those results in a little bit here. And similarly, the other question is, how are these networks evolving with time? For example, as a skill is learned, or as a person goes through childhood to youth to adulthood, how are these networks changing? So taking cue from a lot of the networks literature, some analysis, some basic analysis has been performed in this area in the last decade or so. Some of the things that they noticed, which perhaps are not surprising because we have seen this in other networks as well, are the small world characteristics. What it means is that if you consider this ordered network where every node is connected to its neighboring nodes and all the nodes have very similar degrees, and if you consider this disordered network where edges are placed randomly, the brain network does not look like either of those. It's somewhere in the middle. It's not completely ordered, nor is it completely disordered. Similarly, if you look at the degree distribution, here is a homogeneous network where all nodes have very similar degrees. And here is a completely skewed scenario where one node has a degree of 8, all other nodes have a degree of 1. Now, the brain network does not look like, does not have a homogeneous degree distribution, nor it has a completely skewed distribution. Its distribution is somewhere in the middle. Similarly, they looked at the modularity of the network. They found that there are different types of modules here in the brain network. They also looked at the degree of the several locations. So they computed for every voxel here the number of voxels they are highly correlated with. And the color indicates the degree. For example, here the red regions indicate the nodes that have high degree. They're also called hubs. So from this analysis, one can understand that here at the top of the brain, especially on the right side, there are these high degree nodes. Similarly, in the medial surfaces here, at the rear of the head, which is close to the parietal cortex, these are highly correlated. And they also did the community detection-based analysis where they constructed these voxel-wise networks. I think in this paper, Lee et al. Constructed a 40,000 node network. And then they found clusters or communities, and then they're showing what these communities are here. There are 7 communities that they found, and they associated or they related these communities with the tasks that these nodes or the regions are known to perform. For example, this community shown here in F is the visual cortex. It has to do with perceiving vision and interpreting it. Similarly, there are some networks here that are associated with attention. For example, when a stimulus is presented, these regions are bound to be are known to be related to the stimulus. So the basic network analysis has been done in the last decade, and there are several things that were learned as to where the hubs are, how the communities are, and is the network modular, etc. Now, some analysis has also been done to understand the differences between the resting state brain and the task brain, which means the subject is basically involved in a task. In this particular paper, Hassan and colleagues, they compared two different scenarios. One is resting, the other is the audio. The person is listening to an audio. And here in this analysis, they took a seed location in this particular point here at the middle of the brain, and then they correlated it with all other voxels in the brain in the resting state scan. And they repeated the same analysis in the task scan. So what it is really that they are computing one row in this correlation matrix here, because they are taking a particular region and they are correlating with the rest of the brain. Similarly, they did the same analysis in task, and then they compared the correlations between these two vectors, and then they showed the differences on the surface of the brain here. Now they numbered these different locations that came out to be different. The one that's more that's perhaps larger here is this one shown on the left or outer surface of the brain, which is very close to where the ears are in a person. Now, this region is known to be involved in processing auditory signals, which is perhaps why it is closer to the ears, and it is also seen on the right side near the ears. So the real differences are coming up in where the auditory signal is being processed. So this is a way to basically getting at these regions that are involved in these tasks. Moving on. Fair et al. In 2009, they did this analysis where they had 3 cohorts. The mean age of the first cohort is around 8 years. The mean age of the second cohort is 13 years. The mean age of the third cohort is 25 years. And then they compared the brain networks of these subjects in these 3 different groups. Now here they are showing, so the nodes are shown in these colors and there are these edges, and they are showing this this covered area in blue, which is related to the frontal lobe in the brain. It's at the front part of the brain, which is why it's called the frontal lobe. When the average age is 8 years, all these nodes are relatively interconnected. And when you look at an age group of 13 years, they start segregating into two different groups. As you go from 13 to 25 years, they start segregating much more. This is interesting because the locations are coming from the same or similar regions at the front of the brain. And then over time, they are segregating into different groups and they are not interacting with each other. Now, in a different location, for example, here in these red regions, they are showing the locations that are highlighted here in red. These regions.
Quick question.
Yes.
I'm just trying to understand how one detects these this segmentation that occurs with age. If I were to sort of understand that, is it the case that the same stimulus was given to a person at age 8 and then to 25 and then to 13 and then this fMRI sort of observed which regions of the brain remained active, and I'm assuming that as the person got older, multiple other regions of the brain became active. Is that correct?
So I have to clarify that this is not fast. These scans are obtained while the subjects are resting. So I'm sorry I was not able to clarify it earlier because only this slide was best versus task. This is just when the subjects are resting.
Okay.
And the way they found these communities is by using these evolutionary community detection algorithms that are often used in social networks to see how over time different people form different communities or how their communities change. So they use an approach like that to see how these communities are over these different groups.
Yeah, I guess maybe this can occur if you want to postpone this question. You can. I guess the more sort of interesting question is, you know, what sort of data? I mean, what sort of analysis led to this segmentation? You know, obviously it came from some original analysis of, you know, from the MRI data itself, right?
Mm-hmm. So they were basically looking for communities in each of these different networks. And a community is found like this here, and in the second group, it found 2 different communities. In the third group, it found 3 different communities. The real reason here is that the connections across these 2 regions become sparser and sparser between these 3 groups as you go from left to right. So that resulted in segregating of these groups.
I see.
And in a different analysis here, they looked at the regions from these highlighted regions in red, and then they marked those with these red contours in the 8-year average age group. And then, as you see, with the next group, they tend to come closer, especially because this is a spring embedded layout. If they share more connections, it basically brings those groups together visually. And then in the 25 years age group, it combines all of them into one community. And this is very interesting because the locations are actually separated in space, unlike in the earlier scenario where all the locations are in the frontal region of the brain. Yet, as age progresses, they all form one group where they all are correlated with each other much more than they were earlier. So these are 2 very contrasting examples as to how the different regions in the brain are interacting as the age is increasing with these groups. So this analysis I chose this paper to show how this analysis is done and what sort of messages or the principles are learned about the development of the brain with age. Here in a different analysis, the differences between healthy and diseased subjects is being studied. This is a paper by Linal et al. in 2010 where they looked at these correlation matrices. So these are 15 correlation matrices that are obtained from 15 different healthy volunteers. I believe this was a 6-minute scan from which these correlation matrices were computed. And on the right here, these are 12 correlation matrices that were computed. And the color indicates the correlation between the corresponding regions. Red is high, blue is low. Just by visualizing these matrices, you can see that there are differences from the left side of these plots to the right side, left side cohort to the right side cohort. The left side cohort is relatively more reddish, and the right side is relatively more bluish. This is also shown empirically using this plot down here. The black curve is showing the healthy subjects group, and the red curve is showing the schizophrenia group, and the values on the y-axis are average correlation in these networks. Even though the variance is much higher, you can see that consistently the mean is higher for healthy group than for the schizophrenia group. And this is done at different pre-processing steps. For example, they were using wavelet transformation to compute the time series. This is one of the steps in pre-processing before you compute the correlation. But nevertheless, the consistency with which the connectivity is stronger in healthy subjects is interesting. They also used these networks and studied some properties of these networks. For example, here they looked at the strength of each region. When I say strength, they computed the overall mean of each of the row for each region, and then they compared this mean for this region across different subjects. Here, the x-axis is the region, y-axis is the strength, and the black curve shows the strength of the regions for the healthy subjects. The red curve shows the strength of the regions for the diseased subjects. You can see that consistently the strength is higher for the healthy subjects, and the variance is relatively lower as well. The differences are alarming here. And the other measure they studied was the diversity. And the diversity is quantified as the variance of each row in this matrix. That is, how different are the connections coming out from every location in the brain to the other regions in the brain. And they found that the diversity is higher for schizophrenia subjects than for the healthy subjects. And then they showed which regions have higher strength significantly in between the two groups, and also where the diversity is higher. So I'll quickly, in the interest of time, skip over these and get to some analysis that we did lately, where the goal was to identify the edges in the brain that are different between the two groups. Here in this matrix, these 27 subjects are schizophrenia subjects, the 31 subjects here are the healthy subjects, the columns are the edges, and the values indicate the correlations on these edges. The goal is to identify which edges have significantly stronger correlation in one group over the other. The typical way to do it is to do a standard t-test and then get a significance value, and then rank the edges by the order of the significance value, and then choose the edges that meet the significance criteria. This analysis has been done by about 30 to 40 papers, and recently Peterson, they have done a meta-analysis where they found that the findings from these 30-40 papers do not agree with each other, which means the edges that they are reporting as implicated in schizophrenia do not agree, which means there is a problem here because the observations are not consistent. This could be because the populations that are studied in different studies are different. It could also mean that there is more noise in the data than real signal. It could also be because of the length of the scan that we saw earlier, that if the length is smaller, we don't get reliable estimate of the network. And often these studies use 5 to 8 minute scans, so that's another possibility. Fortunately, we had two different scans from this group that I showed you here in this plot. So this data is coming from the first set of scans. 9 months later, we also got the second set of scans, and all these were resting state scans. And then we repeated this analysis on the both scans because we have the same set of people, and we expect to see similar results because we have the same set of people. And this is what we found. On the x-axis here, I'm showing you the significance of the connection on in the first set of scans. On the y-axis, I'm showing you the significance of connections in the second set of scans. Each circle here is an edge, and if my threshold is 3, there are about 10 connections that are significant in the first scan. There are about 50 connections that are significant in the second scan, and there are only 2 that are agreeing with each other. There are 2 that are reliably significant. And this is much worse because we want to know the connections that can be reliably estimated at least from the same subjects. Otherwise, it will be hard to treat patients or even diagnose patients if they have the disease or not. So here we had some interesting observations that the connections in fact in the brain are not independent. What I mean by that is that if you look at, so here in this plot here I am showing you around 25 connections. These connections look highly similar because in these set of subjects here they are all high, in these subjects they are all low, and again in the control group these subjects they are all low and then they are all high at this part. So by testing each of them independently, especially when there is so much noise, will give us arbitrary results, and so they are barely reliable. So what we did in this analysis was we clustered these edges into groups where the clusters look more homogeneous like this, and then we tested for each cluster, are they significantly different between schizophrenia and control group. In this example, you can see that most of these connections are red in schizophrenia group, and very small fraction of connections are read in the control group, which is the healthy population. And we indeed found that cluster 8, 19, and 24 are significantly different in between these two groups in the first set of scans we had. We repeated this analysis on the second set of scans, and we found that the same three clusters are also significant. Actually, they are the top three in both these data sets. And so, and here are the edges that we found. Here they are connecting from thalamus to the visual regions of the brain. These edges are going from the thalamus to the temporal regions where the ears are in the brain. And these edges are going from thalamus to other regions. So in all these clusters, thalamus is playing a major role. And we try to replicate this on an independent data set where we found that these clusters are also significantly different in schizophrenia and healthy groups. So here by addressing the problem of redundancy and by grouping these clusters, we were able to retrieve the signal even though there was a lot of noise, which a lot of existing methods will have a hard time doing that. Now I want to bring you back to this slide where we talked about how the networks are constructed. Notice that we have been using the entire 6-minute scan to compute the correlations, and then we are treating the resultant matrix as a brain network. By that, we are inherently assuming that the connection is static over this entire duration of the scan. Recently, there was a set of papers by Chang, Handwerker, and Hutchison where they showed that this is in fact not the case. Here in Handwerker's paper, he showed two time series from different locations in the brain, and then he looked at a 32-second interval here. It's called a sliding window because we are going to slide it. For each of this window, a correlation is computed between these two time series, and it is shown here. The 32-second one is in blue here, that starts here, and then the sliding window is shifted right by one time step, and then the correlation is computed and indicated here, and the sliding window is moved all in steps of size 1 all the way to the end of this time series, and the correlations are computed and they are shown here. What you see is that the correlations are changing with time throughout the duration of the scan. For example, here they are very high, there are places where they are very low, and so on. So this paper and other papers that have similar messages have highlighted that we should perhaps look at the brain as a dynamic network and not as a static network. And that raised questions about the validity of the findings in the last decade. And it also raised questions about how do we now take this fMRI data and study the dynamics that are present in this data. Now here are a set of data mining tasks that can be studied, and some of them have been looked at by us and others in this area. The first question is, can we characterize the edges based on the dynamic nature? For example, if this is the correlation, if this is how the correlation changes between two regions with time, can we use how the correlation is changing to characterize the edge? For example, here it's changing relatively more slowly than this particular scenario here where it's changing relatively fastly. Another question could be, can we find the changes in the community structure with time? This is very popular in the social media or the social network literature, where they looked at how the communities are changing with time in different scenarios. Another question could be, can we use the duration for which the two regions of interest are correlated? For example, the doubly sided arrows here, I'm showing you the intervals where the two regions can be correlated on a time axis. So here, let's say the two regions of interest here are correlated in an interval here, here, and here. If you compare that with another scenario where there are many more intervals, one can characterize that these pair of regions in this particular scenario are correlated more often than those in the red scenario shown here. Another related question can be, can we find change points in the network? For example, we can compute these brain networks over time by using the sliding window analysis. And can we identify places in the time axis where the network completely switches to a different form? Now, the changes, the switches can be so evident that the network looks completely dramatically different, like the one shown in this caricature here, or the edges can be very subtle, or the changes can be very subtle, in the sense that a very important edge could be missing, and then that can change the characteristic of a network. So I want to show you a few examples. I'll skip the slide in the interest of time. So this is one example where we studied the intervals where the time series are correlated. In a traditional analysis, if these are the two time series of interest, the sliding window is taken and the correlation is computed and so on for all the sliding windows. And if I'm interested in only those intervals where the correlation is 0.8 or higher, I would select these two intervals here and then say this is where the two regions are highly correlated. But there is a challenge here that we know that window 3 and window 4 satisfy the correlation threshold, but there is no guarantee that the entire interval from 20 all the way to 60 will satisfy my correlation threshold. To overcome this challenge, we came with the notion of maximal correlated intervals, where we guarantee that all the subintervals are also strongly correlated. By that, we can easily model the relationship between two networks with the help of these intervals, where we know for sure that all subintervals will be correlated. Taking this approach, we found some interesting observations. Here, I am showing you the intervals for a pair of regions when the subject is resting and when the subject is shown cartoons. You can see that when the subject is watching cartoons, there are many more intervals where the pair of regions are highly correlated. The doubly sided arrows are showing the time intervals where the time series are highly correlated, and in the case of rest, they are very sparse. And here I am actually showing you the real time series to show how these changes happen. Here they are highly correlated, something happens, they are highly correlated, and then all of these is not correlated at all, and then they become highly correlated. But in watching cartoons, they are more often highly correlated. And then we looked at these two brain regions, and we found out that one of the brain regions interprets color in the object that is being seen. The other brain region here is known to be related to identifying the position of the object in the visual field. For example, when you have to grab something in front of you, there has to be a good representation of where the object is in the visual field so the hand can grasp it. So the region that deals with this particular functionality is interacting with the region that identifies the color of the object, and we presume this is so because the cartoons are being watched here, and to make sense out of these cartoons, both the color and the location where these objects are in the cartoon video are important, and they are being interpreted and they are being used when the subject is watching cartoons. Another interesting scenario we found was that we found, so here I am showing you 6 or 7 different connections in the brain, and each row is showing in black color the intervals where the regions are highly correlated. And all of these 7 pairs are chosen because they all have this total duration of intervals about 700 time points. Now, even though they are all correlated for the same duration, you can see that this particular pair 3-7 has really long correlated intervals. In fact, the smallest correlated interval here, or the shortest correlated interval, is longer than every other interval that is shown in this plot here. This suggests that the pair 3-7 generally interacts for longer periods, whereas the other pairs that are shown here have very short interactions between them. One could use this observation to characterize the nature of this interaction and also to understand what it means or what these regions why is it that these intervals are longer in this scenario. We also found that when we compare the same 3-7 pair between rest and cartoons, we found that these intervals are shorter in cartoons, but they're much more longer in rest. Although we don't really know what exactly it means in terms of the neuroscience principles, this certainly caught our attention. The other kind of analysis we did was we looked for group of brain regions that are interacting in infrequent intervals. What I mean by that is we have designed a frequent pattern mining based framework where we can group the brain regions that are highly correlated with each other only between 4% to 50% of the time. If they are generally correlated in more than 50%, even if we do not do these dynamic brain network analysis, we can still find those relatively easily. But the real information in the dynamics, we believe, is in these short-term interactions between these brain regions. And here I am showing you two different groups of such brain regions. Here these brain regions are in the front of the brain. Here these are in the front as well. There is one at the middle part of the brain. And the green region is the same. So this green region interacts with these three other regions in 4% of the time. And this brain region interacts with these three other regions in another 4% of the time. The very interesting part here is that we did this analysis on data that was obtained at two different time points. So we got an fMRI scan and we analyzed this data. We got an fMRI scan that was collected 9 months later. And we found that in both these scenarios, these combinations are present. The interesting part here is that even though they occur so infrequently, they are very consistent. And similarly, this is another example where there are two regions that are common and they are consistent between the two time scans, even though they occur only in 4% of the time. And this here is another recent analysis by Allen and colleagues in 2012. What they did was they constructed the brain networks for each sliding window. So as shown here, this is one sliding window and that's the corresponding correlation matrix. And they move the sliding window and they compute all the correlation matrices. And they do it for all the subjects in their data, and then they used k-means to cluster these correlation matrices to find 7 different clusters. Here, these clusters are shown in big color, so they call these clusters as brain states. So this is state 1, oops. This is state 1, this is state 2, 3, 4, 5, 6, and 7. You can see that these states have very different signatures. What is shown here is actually the median of the mean of the clusters that k-mean found. So this signature here is quite different from all the other signatures, and it's easy to see that even this one is similar from this one because it's more front dominant, and this one is more in the second half of this plot, and so on. What they also did is they quantified in how many subjects does this state occur at different time points in the duration of the scan. The most interesting plot to me is this one, where over time, this state occurs in more and more subjects. This could be because this state could occur when the subject is perhaps more tired because as the duration of the scan increases, this state is becoming more and more prominent in many subjects. So this analysis lends to very interesting observations to sort of study what the brain states are, to study how these connectivity differences exist, and how do they occur in the duration of the scan, even though this is a resting state scan. This analysis can also be performed in a task scan to understand how the brain switches its state between when the subject is resting to when the subject is shown a video or given an audio stimulus, and so on. So these are several examples of how the dynamics in the brain networks have been studied in the community so far in the last couple of years. And as you can see, these different works are capturing the different aspects of dynamics. There is a lot more to dynamics, and the community is approaching it from different sides, and we are seeing different aspects of dynamics. And there is a relatively long way to go to sort of get a good hold on the dynamics in the brain and to make a complete sense of what it is that we are looking at. In summary, The brain networks are widely studied in neuroimaging, mostly based on the principles that have been observed in social and other related complex networks. The static and dynamic networks are being studied, and then the networks are being compared between scenarios like rest versus task, healthy versus disease, through the development of the subject. And these are very early stages in studying dynamics in the brain, and there is scope for novel data mining approaches to reveal interesting insights into the brain dynamics. And again, as I said earlier, there is a scope here to cross-pollinate ideas between different disciplines, so we can learn from these different disciplines and advance towards solving society's problems. I acknowledge the support I received from my advisor Vipin Kumar, my co-advisor Angus MacDonald, and our collaborator Kelvin Lim throughout the duration of the several projects that we did in brain networks and dynamic brain networks. I also thank my colleagues in various departments, including psychology, psychiatry, and neuroscience. Brent, Andrew, Krista, Ian, and Craig have been very helpful throughout this process. Here are some references. I thank you all for your attention, and I'm open for questions.
So I guess before we get started with questions, since we're at the end of the scheduled time, Gowtham, you've indicated you can stay for a little bit longer. So for those of you that do need to run because you have prior engagements at 1 p.m., please do fill out the seminar survey. That helps us keep our seminar on track and interesting. If you have suggestions for speakers or for topics, please do send them either in the seminar or else send me an email. And I guess with that, maybe we can get questions. So please either send your questions in via chat or ask them if your audio is not muted. Okay. So I guess there is one question that has come in here, and the questioner asks, have you compared the resting state with the state of the brain under meditation?
So there has been some work on this front, mostly that I know from Wisconsin. If there is interest, I can certainly point you to those papers. And recently I was talking to some of our colleagues who were collecting this data from people who meditate regularly versus from people who do not meditate at all, and so on. So there have been a number of studies on this front. I don't recollect the papers on top of my head, but I can certainly point you to those if you could shoot me an email.
Very good. I guess one question while we're waiting for people to send questions is the following. I guess only recently is sort of the appearance of data mining techniques and applying them to sort of these domains. What kind of analysis was done prior to emergence of these data mining techniques?
That's a very good question. So the overview that I gave here in this talk covers mainly the analysis that was done by modeling the brain as a network in fMRI data. Even before that, a lot of analysis has been done using linear models. For example, when a task scan is obtained, so the task will have a certain time series. That is, when the task is on versus when it is off through the duration of the scan. The task could be on for 30 seconds, the subject could be resting for the next 30 seconds, and the task can be shown for the next 30 seconds, and so on. So this time series is often is used as a target, and to identify the locations that are responding in a very similar way. So a linear model was often used to understand which of the regions are active that are corresponding to this task. So that was one common theme. The other big area is called independent component analysis. These have been widely used to study both resting state and the task data. They are called independent components because this is actually I believe came out of electrical engineering, and the motivation was that there are 4 mics in a room and there is a party going on, and the speakers are talking, and everybody is equivalent to the receiver. And what the receivers hear is a combination of the audio signals from these 4 different sources. And the goal of the independent component analysis is to tease apart these 4 different original sources, because all the participants will hear a combination of the 4. So the goal was to separate out the 4 different original sources. This was originally applied to EEG data, where the data is more diffuse, in the sense that if the the data was not spatially as high in resolution as it is in fMRI. So it used to, so the recordings from EEG are often a combination of the original sources. So that was used originally in EEG and later on adopted in fMRI, and that used to be another popular technique before the brain was modeled as a network. Actually, only in the last decade or so, the brain networks became popular. I mean, the network construction and analysis methodology has become relatively more popular.
Thank you. There's a question online, and the questioner asks, how have your data mining studies with neuroimaging data, are they actually being utilized in any studies at hospitals, doctors, etc.?
So, as I said earlier, in the case where there were these, the issue where we had 20 years of fMRI from Neuroimage, one of the dissatisfactions mentioned in those reviews is that unfortunately fMRI-based results are not being used in clinical practice because the signal that we get here is so little that it cannot be reliably used for clinical practice. For it to be used in clinical practice, the signal has to be very high and the reliability has to be really good. Because of the nature of the data, and most of these are ongoing projects and analysis, and typically we study 30 subjects in disease and healthy and so on. So these are still early stages, I would say, before the real outcome can be used in clinical practice. But as of now, based on these reviews, I don't think fMRI-based results are being used in clinical practice.
Okay. One more questioner asks, where do you see your future research going, and what would make it much more applicable to sort of nuclear medicine?
So one main theme that I'm excited about is that in some of these intervals that I was showing you, so I was showing you that these intervals are being computed from resting state versus task scan. We recently have access to this data where in the same task scan, for the first 30 seconds video is shown, and the next 30 seconds the subject is rested, and the next 30 seconds another video is shown and rested, and so on for the duration of 8-minute scan. And we are applying this technique to sort of see if these intervals match well with either the resting state scenario or with the task scenario, and we are seeing very interesting results. And so there are many of these different types of these task experiments that are conducted, and studying dynamics to understand which regions collaborate to sort of achieve a task is where I see going forward, because we have a lot more context in task because we can associate the interaction with the task at hand. So that's something that I'm excited about. Other than that, we also get these data from, for example, recently I've been looking at a scenario where we have data from alcoholic subjects. Some of them have relapsed and some of them have not. We are trying to understand how the network has changed after this treatment, due to which some of them have not relapsed. So to sort of able to determine the effectiveness of the treatment methodology. So there are a number of interesting questions in this area, and I'm trying to understand how these data mining techniques can help solve those, answer those questions, and also build new techniques because of the scale of the data and the number of challenges, as we saw earlier, that there is this multi-level resolution. Do we use bigger regions or smaller regions? Should we get longer scans or smaller scans? How does that impact our results? And how can we build these robust data mining tools to be able to discover signals despite these challenges?
Any more questions, folks? While I'm waiting for one, Gowtham, I have one question. You guys have, it's a fascinating talk. You've studied sort of diseased patients and sort of how the brain function differs from healthy patients. One very intriguing factor about the brain is its ability to repair itself, which is rather remarkable. So there are people who have, have some diseases, and then over time actually have brain function that recovers, especially, for example, stroke patients, right? Does that offer any particular line of inquiry for your research?
That's interesting. At the same time, it's to me more biological as well, because the regions that receive stroke is mainly have loss in the cell and neuron tissue there. So it's certainly interesting to see how or what drives this recovery, and how what we learn from this can be used in other cases, especially in Alzheimer's, because there is a lot of loss of the white matter connectivity and eventually the gray matter connectivity, and any understanding that we have about this recovery can be potentially used there.
Okay, thanks. So, folks, any further questions? Otherwise, we can, thank Dr. Gowtham Atluri for a great talk. But let's wait for a second. Any further questions? Okay, hearing none, let's do the following. I guess first thing, our next seminar is on September 18th, where Dr. Dimitris Papilopoulos is going to be talking about big graph analytics through low rank approximations. He's currently at UT Austin. So that's going to be on September 18th, Friday at noon Pacific. Hope you all can attend. As I mentioned earlier, please do also fill out the seminar survey, provide ideas for speakers, provide your feedback for what we could, how we could make this series of events more, valuable to you guys. And as in previous practice, please, I'm going to unmute all of you. If you have muted yourself, please unmute yourself, and let's all extend Dr. Atluri
and also check.
Thank you, everyone.
Thank you. With that, Gowtham, thank you very much. It's a wrap.
Thank you, Sridhar. Thank you so much.
Yeah, thank you.